Good evening, students. So, in this session, we are going to deal with the last bit of sex determination. That's environmental and hormonal effects on sex determination, and the final bit, that is amniocentesis. So, we'll deal with the topic. So, first one, we'll see the how the environment will affect on the determination of sex. So environmental sex determination is the establishment of sex by a non-genetic cue, or you can say signals such as nutrient availability and experienced within a discrete period after fertilization, especially. Okay, and environmental factors which often influence, especially sex determination during development, or you can say sexual maturation, include some of the natural calamities, that is light intensity, photo period, temperature, nutrient availability, and pheromones. Okay, so which are emitted by surrounding plants or animals also. Okay, so this is an in contrast. Okay, this is an in contrast to genotypic sex determination, which establishes sex at fertilization. Okay, which expresses sex at fertilization. So by genetic factors such as specialized charactered things, that is nothing but a sex chromosomes. Okay, under true environmental sex determination, once sex is determined, it is fixed and cannot be switched again. Okay, that's an important thing, that cannot be switched again. So environmental sex determination is different to some forms of some, you can say, the subsequential, or you can say sequentials also, hermaphroditism. Okay, so in which the sex determined as a flexibility, or flexibly after fertilization throughout the organism's life. Okay, so in that, uh, some of the important factors which affects on environmental determinations. First one is the adaptive significance. The how the adaptations will significant in sexual determination through environment. So environmental sex determination is similar to certain forms, okay, certain forms of sexual selection, in that there are oftentimes different and opposing selective pressures on males and females. Okay, so especially because of cost of reproduction. Okay, so that we are calling it as a cost of reproduction. So that is sexual selection. So it is a common throughout the most of the birds also. Okay, and often resulting in sexual dimorphism. So and we can say our size and appearance and differences between sexes in the same species. So in environmental sex determination, the selective pressures, okay, over evolutionary time. Okay, selective pressures for evolutionary time have selected for flexibility in sex determination to optimize fitness in a heterogeneous environment. So this is because of mainly the different costs of sex in males and females also. So certain environmental conditions differentially affect each sex. Okay, and in such that it would be beneficial to become one sex. Okay, and not the other, the same species. So this is especially you can say pertinent for sessile organisms mainly. Okay, and that cannot move to a different environment. So in plants, for example, we can say, take some examples. Female sexual function is often more energetically expensive, because once fertilized, there must be significant stored energy to produce the plant byproducts. Okay, and next will be the mechanisms, how the genetical helping some of these mechanisms. Okay, so there is a lacking genetic information coding for separate sexes, such as sex chromosomes, especially individuals that exhibit environmental sex determination contain genetic information coding for both sexes. Okay, both sexes, especially on autosomes also. So in general, so once exposed to certain environmental causes, or we can say signals, epigenetic changes. Okay, epigenetic changes means the without altering the DNA sequences, some alterations have been uh, happened. Okay, so we are currently epigenetic changes cause developing individuals to become either male or female. Okay, environmental signals that often trigger the development of males or females include especially the temperature and food in case of especially animals, and also water availability and photo periods and competitive stress, and also especially the pheromones from the specific individuals. Okay, the specific mechanisms and signals vary in between species also. Okay, that can also matter. And then in different organisms, okay, different species. Okay, so how these can be explained means, first one you can say crustaceans. In crustaceans, the amphipod of crustacean, that is example here taken is the gammarus and dubin. Okay, it produces some males early in the mating season and females later. Okay, so in response to the length of daylight of the photo period, especially. So because male fitness improves more than female fitness with increased size, environmental sex determination is adoptive in this system. Okay, actually by permitting males to experience a longer growing season than females. Okay, and another one is the brachipod crustacean, that is Daphnia magnum. So it is parthenogenetically produced male progeny. Okay, so there is an undeveloped egg producing male progeny in response to a combination of three environmental factors. That is by autumn. Okay, there is a shortage of food and rise in population density, etc. And in case of annelids, okay, one of the best example is Bonelia viridis. Okay, so this in detail we are seeing in this session only later. So it is a marine worm has location dependent sex determination. Okay, so the sex depends on especially where the larva lands. Okay, so we'll see in this session only later. And coming to the vertebrates. Okay, the sex of most amniote vertebrates, that is amniotic fluids, amniote vertebrates such as mammals and birds. So it is determined genetically. Okay, but however, some reptiles have a temperature dependent sex determination. So their sex is permanently determined by thermal conditions experienced during the middle third of embryonic development. Okay, so the sex of crocodilians and especially the spinodontians is exclusively determined by temperature, especially. So, in contrast, um, some of the squamates, that is lizards and snakes, and also turtles, they exhibit both genotypic sex determination and also temperature dependent sex determination also. Okay, so although the temperature dependence is much more common in turtles, okay, than in squamates. So in most turtle species, especially in turtle species, um, only females produced at a high temperature, that is 30 to 35 degrees Celsius, and only males are produced at a low temperature. Okay, so that's in between 23 to 28 degrees Celsius. Okay, the sex ratio changes significantly from all male to all females over just a 2 degrees Celsius change in temperature. Okay, and the reverse is true in many crocodilians. Okay, so it is reverse in crocodiles and also alligators and also some lizards. Okay, so where males are produced at a high temperature and females are produced at low temperature. Okay, and in snapping turtles, the species, the snapping turtles, and in some other turtles as well as example taken here is Australian crocodile. Okay, females are produced um, at high, that is a 30 to 35 degree, and also in 25 degrees Celsius also. But males are produced in especially intermediate temperatures. Okay, in between 25 to 30. So intermediate temperature. That means high temperature leads to faster growth, shorter incubation periods, and larger juvenile size, and hence in a larger adult size also. So therefore, as an adaptation, it has been observed that the sex which develops at a high temperature is larger in especially body size, okay, and exhibiting sexual dimorphism. So, for example, in most of the turtle species only, the females are bigger in size and at a high temperature, mainly females are produced. On the other hand, so in crocodilians and in some lizards, males are bigger and they are the ones produced at a high temperature, especially. Okay, the individuals where females are produced both at a high and low temperature also, and males are producing intermediate temperature. Okay, they are related to turtles where at a high temperature females are produced, but their eggs face wide on unpredictable range of temperatures. Okay, so either it may be due to a geographical change also, or due to a large temperature gradient in particular their nest also. Okay, under these circumstances, the above pattern will ensure, okay, so this pattern will ensure a reasonable proportion of females in all environment also. Okay, so this is the environmental changes, okay, on some of the things which are responsible for the changes of sex gene. Okay, and then coming to the hormonal controls. Okay, that means the general theory of sex, okay, as the result of balance between the opposed tendencies, so has been successively applied to the conditions, okay, found in many animals, especially. So in the higher animals and especially in vertebrates, the secondary products of the sex glands, okay, and the sex hormones may very considerably alter the final character, especially. Okay, so a large number of uh, case or non-aware sex is modified due to hormones secreted from especially sex organs. Okay, so three of the classical examples are there. Okay, so hormonal or environmental control, you can say, of sex could be seen here. So the first one is sex in Bonelia, and next is the Crews hen, and the free martin. Okay, these three are the important in hormonal effects of sex determination. So first one is sex in Bonelia. Okay, so in Bonelia viridis, okay, Bonelia viridis, it is a marine worm. Okay, all larvae are genetically and cytologically similar here. So in this worm, a male individual lives in the uterus of female. Okay, that's a interesting thing. Here you can see the male lives in the uterus of female. So if a particular larva settles near proboscis, okay, near proboscis of an adult female, it become a male. Okay, it become a male individual and lives in the uterus of the female. On the other hand, so if it has to develop free in water, it will become a female. Okay, it will become female. So similarly, if a partly developed male is detached from the proboscis, it will become a intersex. Okay, so obviously the proboscis secretes a substance suppressing the femaleness, especially. Okay, so this is one of the best example of sex in Bonelia. Okay, we can see this picture. This is the adult worm. Okay, so if the male is attached to the proboscis, okay, and if it is growing in a uterus, that will become a male. Okay, if the worm is freely living in water, then it will become a female. So then next will be the cruise hen. Okay, so this is the case of a complete sex reversal. Okay, that is complete sex reversal. So which was reported in 1923 by scientist Crew. Okay, so where a fertile female fowl, that is hen, which had already produced offspring, changed over to a fully fertile male. Okay, so you can say cock. So this resulted due to a damaged ovary in the female, especially. Okay, so it is believed that ovary in female secreted a male suppressing hormone. So therefore, in absence of ovary, testis could be could develop. Okay, so this is the crew hens. And next third one is the free martin. So free martin means, so it is the scientist name. Okay, so in this case, especially in a cattle, when twins are produced, so one of them being a male and the other will be a female, the female will become a sterile and male will become a normal. Okay, so these are known as a free Martin. Okay, so after his the worker name, okay, name of the worker, and would be produced only when there is a vascular connections. Okay, then there will be a vascular connection between two embryos. Okay, the hormone perhaps suppress the development of ovary in a female, so that the female will become sterile and the male will become a normal. Okay, so this is all about environmental and the hormonal effects. Okay, on sex determination. So then we'll move to the next bit. That is the last one. It is amniocentesis. As you all know, amniotic fluids. Okay, where the fetus is growing. So amniocentesis, it is a procedure used to detect chromosomal abnormalities, okay, especially in a developing fetus. So amniocentesis is a procedure in which amniotic fluid is removed from the uterus for testing, or we can say treatment also. And amniotic fluid is a fluid that surrounds and protects a baby during pregnancy, as you all know. Okay, so this fluid contains fetal cells and also proteins. So amniocentesis can be done for several regions. Okay, so some of regions are listed here. So first one is the genetic testing. Okay, if the condition suspecting for Down syndrome, okay, so that can be studied by taking a sample of amniotic fluid. And then fetal lung testing. Okay, that means the maturation of lungs. Okay, so that can be enough for taking the birth. That can be decided by fetal lung testing and by taking the sample of amniotic fluid. And also diagnosis of fetal infection. Okay, if any abnormal conditions happening to the fetus, that means it may be anemia or erythroblastus fetalis, that is a rh sensitization of, so as you studied, so that can be studied by taking the samples, okay, of amniotic fluid. And then it can be a treatment purpose also. That means if too much of amniotic fluid is accumulated in the during pregnancy, so that can be helping for a draining of that excess fluids. And then it can be helping for paternity testing also. That means this amniotic fluid contains some cells, as I said, no. Okay, so by that DNA is collected from that fetus, and that can be compared to DNA from the potential father. Okay, so another important thing is the genetic amniocentesis. Okay, so this is the special case, genetic amniocentesis, and it can be information is about the baby's genetic makeup. Okay, how the genetically. So generally, the genetic amniocentesis is offered. Okay, it is offered when test results might have significantly impact the pregnancy, whether the pregnancy should be continued or it can taking a decision to stop. Okay, if the abnormality is there. Okay, and genetical amniocentesis is usually done between 15 to 20 weeks of pregnancy. Okay, if it is done before 15th week, so it may become leading to complications also. So the test that the genetic amniocentesis might considered, okay, when if the positive results from prenatal screening test, okay, that is the first trimester, the first 3 months of pregnancy. Okay, some of the hormonal testings will be happening. Okay, so that will be done. So that is called a screening test. Okay, if any abnormal conditions were diagnosed, okay, that is a prenatal cell-free DNA screening. Okay, if any positives or worrisome is happening, so that may be resulting to take off the genetic amniocentesis. And the chromosomal condition or a neural tube defect in a previous pregnancy. Okay, so if the previous pregnancy was affected, okay, by a condition that is Down syndrome or any abnormal genetical syndromes, and also neural tube defect, so which is a condition affecting baby's brain, okay, and also spinal cord. So where spinal cord is does not end at the end of spinal cord, okay, and they have this. If conditions happens, then the health provider might suggest these amniocentesis to take on. And if the pregnancy age of a female is about 35 or plus, okay, so this is the condition where the woman of 35 years or older will be a higher risk of chromosomal conditions. Okay, so especially the Down syndrome. Okay, so when this is happening, then the doctors suggest for this test. And the if a family history of a specific genetic condition, okay, that means any one partner is known as a carrier of a genetic condition. Okay, so we have studied some of the conditions. If the any one partner is a carrier, okay, so that may be a chance of taking this test. Okay, so as I said, the spina bifida, it is a neural tube defects. Okay, so by these conditions, amniocentesis can be used to diagnose these all things. And also abnormal ultrasound findings. Okay, so if in a month of trimester, okay, so the ultrasound findings helps to diagnose any abnormal conditions. Okay, so that will be ruled out genetic conditions associated with the especially abnormal ultrasound findings. It may be a heartbeat, or it may be a lungs test, it may be kidney test. Okay, that will be reasoning for these genetic amniocentesis. Then how the procedure takes place. Okay, in amniocentesis, a hollow needle is inserted through the mother's abdomen, okay, and into uterus, and amniotic fluid is drawn from the for analysis. Okay, so this procedure involves a hollow needle is inserted through the abdomen of pregnant women to withdraw a small sample of amniotic fluid from inside the amniotic sac. Okay, from inside the amniotic sac. The physician used to ultrasound to ensure that needle does not touch the fetus. Okay, so the withdrawn sample of amniotic fluid contains specially free floating cells, and that cells later isolated, and these sample then grown in a petri dish in a lab, okay, for a 1 week, and then that cells can be studied under a microscope. And especially the karyotyping is studied here. It is the arrangement of chromosomes. Okay, what you are going to do in a laboratory, that is a karyotype arrangement of chromosomes. So that will be done here. So to ensure that the proper chromosomes are developing, okay, proper size of chromosomes, okay, starting from chromosome number 1 and up to the sex chromosomes also. Okay, and here some risk factor is also there. Okay, that means which include some as a side effects like crampings, bleeding, infections, and you can say preterm labors, and also some case miscarriage also happens. Okay, but the accuracy of this test is about 99.4%. Okay, and but less than 1% may suffer from the miscarriage also. And if considering and getting amniocentesis, but one should talk before a doctor, okay, so that the conditions can be studied well. Okay, so this is all about the things, that is environmental and hormonal effects on sex determination and also amniocentesis. Thank you.